Aloha everyone, and welcome back to Biology 101 here at Chaminade University. Today we'll be delving into Unit 8, or Patterns of Inheritance. We'll be talking about all sorts of types of inheritance patterns. We'll be talking about the physical basis of inheritance and how we discovered the basic traits of inheritance, and how traits are inherited if they are inherited by themselves, or how traits are inherited as a group, so multiple traits. We'll also talk about Mendelian inheritance. We'll then talk about exceptions to Mendelian inheritance, talk about gene linkage, or genes that are located on the same chromosome and how they might or might not travel together. And we'll talk about genes that are on the sex chromosomes. So we'll talk about sex-linked genes and how that affects men differently than women. And then we'll talk about multiple different types of human genetic disorders. But first, let's delve into some definitions. So inheritance is just the process by which the traits of organisms are passed on from parents to offspring, and it's why you and your siblings resemble your parents and why they resemble their parents, etc. Now, genes are, as you may remember, we talked about DNA briefly when we talked about biomolecules. So genes are comprised of DNA, so they're sequences of nucleotides, which are located at specific locations on chromosomes. So gene is going to code for a specific information. A hereditary unit could code for a protein, etc. And a gene is going to be any unit of hereditary that encodes information needed to produce everything that makes you, you. And genes can be thousands of segments long, so they comprise multiple segments of DNA, and they can range from hundreds of bases to thousands of nucleotides in length. And they're located in a specific location within the genome. So just the way a library book would be in a specific location in the library, if you were to try to go ahead and find it, the same thing with a specific gene, it would be located in a specific region within the chromosome or the genetic library. Now, as you may recall, you're going to carry one copy of each chromosome from each of your parents. And that means that you may or may not have identical alleles at particular locations on your genes. So if you have the same kind of gene with the same characteristics, then it's considered homologous. That means that they're going to carry the similar genes. That doesn't necessarily mean that it's going to carry the exact same alleles, right? You might have different alleles from your mom and different alleles from your dad, but it's going to have, for example, chromosome 1 is going to have the same eye color, even if it's blue eyes and brown eyes. So genes for the same characteristic are going to be found again at that same location or the same loci, but that doesn't mean that they're necessarily identical. We can have alternative forms of the genes that are found at the same locus. Again, these are called alleles. And alleles originally arose as mutations. Mutations are spontaneous changes in the nucleotide sequence of the genes. And most of those mutations are going to be in our somatic cells, so cells that aren't going to get passed on to our offspring. However, every once in a while a mutation is found in a sperm or an egg, and then that mutation will be passed on from parent to offspring. Most mutations that are in your DNA originally appeared in the reproductive cells. Again, anything that was in your somatic cells would have been lost and not actually made it on from parent to offspring. And every once in a while, mutations arise in the reproductive cells of the organism's parents. But again, this is quite a rarity. Usually it's just going to be passed on from those particular cells. And as we mentioned, at any given locus, you're going to have two alleles. Those alleles may or may not be the same, right? So if you have the exact same allele at the exact same loci on two different chromosomes, so both of your chromosome 2 carry the blue-eyed gene, for example, that's considered to be homozygous. If, however, on chromosome 2 from your mom you have blue, and on chromosome 2 from your dad you have brown, then you'd be considered heterozygous at that locus, or a hybrid, right? You have a combination of genes from both parents, and this is good because this gives you genetic variety, which might help you rise and overcome whatever genetic challenges you were exposed to in your environment. But I digress. So this is what I'm talking about. So these two are homologous chromosomes. For example, they're both chromosome 1, or they're both chromosome 7, or whatever, right? But within chromosome 7, there are specific locations where the genes are supposed to be located. So these are genes for, as we mentioned, eye color, hair color, etc. And if you have the exact same, then it's considered homozygous at that locus. So you have 2 for brown eyes, etc., 2 for blonde hair. But if you have different genes at that locus, then it's considered to be heterozygous. So you have a different allelic form here than you do here. Now again, these are both going to be for the same gene. They're just different versions of the same gene. Now let's talk about Gregor Mendel for a little bit. Gregor Mendel was a monk. He's from Austria, and he actually was very fastidious about how he studied his work. So he looked mathematically at inheritance patterns, looking at different genes, different alleles, and the distribution of these alleles in both the gametes and the zygotes during sexual reproduction. And because he was a monk, he actually he was told that he had to use something that they were able to eat because it had to be practical, and so he used the pea plant, which then they could eat the leftovers, which then it took place in the garden at the monastery. And he had a fantastic scientific background, and that allowed him, and a mathematical background, that allowed him to look at the patterns in the inheritance characteristics. So this is Gregor Mendel. And again, he crossed pea plants and noted the particular traits and noted the percentage of those traits in the parental and then in the offspring, etc. Looked for emergence patterns and when those genes would reemerge, those expression patterns would reemerge. Again, he didn't know what the word genes was. He was calling them inheritance units, but he was looking for the patterns in the offspring and then the offspring and the offspring of those offspring, right? And Mendel actually had a fantastic scientific method. So one of the key secrets to Mendel's success is that not only did he choose the right organism, so his organism had a quick reproductive time and had lots of offspring, so his numbers could get up pretty high, which meant his statistics were pretty good. Also, he had very specific precision with the experimental design and experimental performance, and he was fastidious about analyzing the data. So pea plants actually make really good organism for inheritance studies because they have stamens, which are the male structures, and those are going to produce pollen, and then inside the pollen they're going to have sperm. Sperm are going to be male gametes, right? And pollen are basically the vehicle for those gametes. And then the pea flowers also have carpels, which are female structures, and they have the ovaries, which produce the eggs. So they do have sperm and eggs, and they are going to end up undergoing sexual reproduction. And the pea flowers have petals that enclose the male parts and the female parts. So that's going to prevent entry of pollen from another pea plant unless you actually pollinate them yourselves or you allow them to self-pollinate so they can pollinate at a particular time. So that meant that Mendel could he could take, see, we have all of our eggs and our sperm, they are housed inside here and they are separated, and that would prevent them from fertilizing. So he can actually open them up and dissect them out and separate out the female carpel here, which produces the eggs, and the stamens of the male, and cross them however he chose. And it would also prevent cross-fertilization, because if he was to remove this part and fertilize, etc., then he would make it that he couldn't fertilize with other plants, right? So he would be very specific about which sperm fertilized which eggs, and he had fantastic note-taking skills. Now, as I mentioned, they do naturally self-fertilize. So that means that pollen from the plant transfers to the same plant where the sperm fertilizes the plant's egg. So they do have auto-fertilization events that can occur, and he was able to then also cross-fertilize. So he could do it two different ways. He could do self-fertilization if he wanted to just see what happens in 2 generations, for example, or he could cross-fertilize, and that would mean that he would take pollen from selected plants and dust the female parts of other plants to be able to create his own offspring, right? Now, he had a very simple experimental design. Instead of looking at a bunch of different things, he was looking at individual characteristics one at a time. He identified these characteristics as traits, and he made sure that they had unmistakable different forms. So there was no sliding variability. He didn't look at anything that was on a sliding scale. He looked at things like white versus purple, right? And he individually studied one trait, and then he individually studied multiple different traits, and then he eventually started studying the multiple different traits kind of together and seeing how they correlated. And we'll get to that in a minute. But originally he started out studying one trait at a time. And he watched the inheritance of these traits, right, these inheritance units, he called them, for multiple generations. And he counted each of the offspring with each of the different types of traits. So it was very black and white, basically. Either it was white or it was purple when he was looking at flower color, etc. And he started to look at basic patterns of inheritance and watching them emerge. He also used different terminology, and as I talk about it, I'm going to tell you what kind of terminology we use versus the terminology that he used. So he used the terminology true breeding. Now remember, he didn't understand genes yet. He was the person who started creating the math that allowed us to understand genes. So he didn't understand at that point what genes were, but he did know that he had organisms that possessed traits that remain unchanged by all offspring produced by self-fertilization. So this entire lineage is always going to produce white pea plants, for example. So he called them true breeding. We would now call them homologous recessive, and what that means is that we know now that they have 2 copies of the white gene, or white flower gene, which would make it true breeding, in that it would never end up with anything other than white offspring. Then he also performed cross-fertilization experiments of the pea plants using those true breeding organisms. So he would take a true breeding white and a true breeding purple. And again, we would call that homozygous white and homozygous purple. And the parents of that cross are going to be demonstrated as uppercase P. And the offspring of the parental generation are going to be the filial generation. That's what F stands for, is filial. And then the offspring of the F1 generation or the F2 generation. And then, so this is what he saw. Basically, he took a true breeding, again, homozygous recessive white plant with the true breeding. This would be a homozygous dominant, and that's only because purple is dominant to white, which would be recessive. So this would be homozygous dominant and homozygous recessive. We put them together and we end up with all purple flowers. So the first generation is comprised of all purple flowers. So some scientists might look at that and say, oh, the white gene was just swallowed up, or the white color was just all swallowed up, and then they wouldn't bother to push it any further. But if you take this all purple plant, it actually turns out that this all purple flowered plant is significantly different from this all purple plant, and I'll talk about why in just a second. All right, so if you take that F1 generation and allow it to self-fertilize, in the F2 generation, you end up with 3/4 purple and 1/4 white. So all of a sudden, the white trait reemerges. So that indicated the white trait was not didn't disappear; it was simply hidden in the background. So then, when Mendel self-fertilized the F2 generation, the F3 generation, all the white-flowered plants, produced white-flowered offspring. And so he was able to get back to the true breeding, which really, again, as we know, is homozygous recessive. And in fact, in order to express the white trait, it would have to be homozygous recessive, so we now know that. He didn't know that at the time. Again, he was just looking at the math. And when he's looking at the white, all of them were, again, what we call homozygous. They were homozygous recessive, or he was calling them true breeding. But if you look at the purple, right, again, we have 4 different types. Well, we have 3/4 are going to be purple and 1/4 is white. But if you subtract that white out and you're only looking at purple, now we're looking at thirds, right? 1/3 were true breeding and 2/3 were hybrids. And those hybrids could then pop out white flowered offspring. Again, at that same ratio of 3 purple and 1 white. And so this was starting to become really interesting to him. Not only did it disappear, but then it would hide and then pop back up in other generations. And so he started questioning how that worked. But let me walk you through what I just told you with pictures if you're a visual learner. So here, that first generation offspring, and remember the first generation is going to be the cross of true breeding purple and true breeding white. That gives us these guys here. And if they self-fertilize, that means they cross with each other, we get 1/4 that are white. And these white are what he considered to be true breeders. From this point forward, they were only going to produce white if they cross-fertilized. I'm sorry, if they self-fertilized. Or if they, I guess, fertilized white plants. On the other hand, we have these 3/4 that are purple. Right now, if we look at those 3/4 that are purple, now we're looking at them in 1/3, right? So one of them was going to be a true breeder, and 2 of them were what we now know to be heterozygate, meaning that they actually carried that white allele hidden within their genotype. All right, so if we look at these inheritance patterns of Mendel's crosses, we have a pretty simple explanation. Well, a 5-part explanation anyway. The first is that each trait is determined by pairs of genes, and we talked about that previously. At any given locus, could have 2 alleles, right? And, well, 1 allele for each of the different chromosomes, but we have 2 different copies of the loci. That means that we can either be homozygous or heterozygous. And if the true breeding white flower plants have different alleles than the true breeding purple flower plants, they would both be able to present different phenotypes because they have different genes, right? And if they're true breeding, that means that they're both homozygous. So we have homozygous dominant and homozygous recessive. Now, if we have 2 different alleles present, that means the heterozygote, when we have 1 purple allele and 1 white allele, the dominant allele masks the recessive allele, even though that recessive allele is still present. So in that heterozygote individual, they're going to appear to be purple, but they're going to have 1 purple and 1 white gene. So in this edible pea plant, the purple flower trait is dominant to the white flower trait. And these pairs of alleles have to segregate independently, right? That means that they're going to split back up during meiosis. Again, Mendel didn't understand meiosis, but he did make the first law of segregation, which led us to understand how meiosis works. And what that means is that they're going to random chance segregate into one gamete or another, right? So random chance determines which allele goes to which gamete, which means that these alleles are going to separate separately, and that means that the distribution of alleles to the actual gametes is also random. And true breeding organisms, again, he called them true breeding, we're going to call them homozygote. They have 2 copies of the same allele, and they can either be homozygous dominant or homozygous recessive, right? So 2 of the purple or 2 of the white. And organisms that are hybrid or heterozygous have 2 different alleles at that given gene, right? 1 white and 1 purple. So if we have a homozygous parent, and that means that that parent has the exact same copy at either one of these gene loci, then in the gametes are going to be the same in terms of that allele. They're only going to be able to produce this capital A allele. But a heterozygote parent is able to produce gametes that vary, right? One that's going to be capital A and one that's denoted here as lowercase a. And this could be the purple and white that we were just talking about, right? So a heterozygous plant, or heterozygous parent is going to be able to make gametes that are either purple or white in terms of what gene they're carrying. Okay, so that's going to lead us to something called a Punnet square in just a minute. But before we talk about the Punnet square, I want to kind of walk you through it. So P is going to be a capital or lowercase. The capital P is going to, in this case, stand for purple, right? So capital P for purple. Now, in all cases, when we talk about homozygous dominant and homozygous recessive relationships, the dominant is always going to be capitalized and the recessive is always going to be the same letter as the dominant. So we're not going to see lowercase w, right? It's going to still be P, but it's going to be lowercase. Right, so the white flower is going to be recessive and it's going to have the same letter, in this case P for purple, as the dominant, but it's going to be lowercase. And again, a homozygous recessive is only going to produce the homozygous recessive gametes. And this one who's homozygous dominant is only going to produce the homozygous dominant gametes. All right, so if we cross the purple flowered plant and the white flowered plant, again, now we're looking at it in terms of what's happening to the actual alleles. That's going to produce all purple flower offspring because all of them are going to get 1 large p and 1 lowercase p, right? Means they're all going to have this intermediate genotype, heterozygous genotype, that's going to produce the white phenotype. I'm sorry, it's going to produce the purple phenotype. Now, the dominant P, right, again, that's going to be the capital P, and that's going to be purple gametes from the purple flower plants combined with recessive P gametes from the white plants are going to produce hybrid purple flowered plants. So all of the heterozygote are going to look like this. That means they're all going to appear to be purple. All right, so the F1 offspring are all heterozygotes. That means they each have 1 copy. They all have 1 copy of the dominant and 1 copy of the recessive allele for flower color. And when they're allowed to self-fertilize, they can make gametes in the form of sperm and eggs that can be produced in a lot of different ways, right? So a heterozygous plant produces equal numbers of dominant and equal numbers of recessive sperm, and again, equal numbers of dominant and recessive carrying eggs. And so when they self-fertilize, each type of sperm has an equal chance of fertilizing each of the type of eggs. And we have these types of genotypes that can be produced from that. And I'll walk you through how this happens, but I promise you, and we'll get there in just a minute, that that's going to turn out to be a ratio of 1 to 2 to 1, or 1/4, 1/2 to 1/4. 1/4 of them are going to be homozygous dominant. 1/2 are going to be heterozygous, meaning they have 1 dominant and 1 recessive allele, and 1/4 are going to be homozygous recessive. And if you have the way that you combine these alleles, that's going to be called your genotype. So this here would be a homozygous dominant genotype, and this would be a heterozygote genotype. However, the phenotype is the outward appearance, and both of these, even though they are different genotypically, appear to be purple on the outside. So both of those are going to have the same phenotypic expression. All right, so I'm going to bring you back now to where we were just talking about that first cross, right? So here we have the purple parent that has the dominant, homozygous dominant, so 2 copies of the dominant allele. The only sperm and eggs that they can create are going to have the purple allele. Same thing with the homozygous recessive, only going to be able to produce the white allele. Now, if we cross these, we can end up, and it doesn't matter which way, whether the sperm is carrying the purple and the egg is carrying the white, or whether the sperm is carrying the white and the egg is carrying the purple, either way, the combination of a dominant and a recessive allele, or a recessive and a dominant allele, is going to come out with an offspring that's going to have a purple phenotype. Again, phenotype being an outward appearance, even though they are heterozygote. So the genotype is heterozygote, but the phenotype is purple. All right, so if we fuse the gametes from the F1 generation, we have 4 different combinations that can happen. We can end up with 2 dominant alleles. That gives us the homozygous dominant genotype and a purple phenotype. We can end up with a combination of purple and white in 2 different ways, right? From the sperm and the egg, or from the egg and the sperm. Either way, that's going to give us the heterozygote genotype and the purple phenotype. And we can also have the recessive alleles come together, giving us a homozygous recessive genotype and a white phenotype. Now, does this look familiar, right? This was the 3-to-1 that we talked about earlier, but what I said was that 2 of them had a distinctive difference from the third if we were looking at the purples, and that's because this purple is what Mendel would have called a true breeder, but is a homozygous, whereas these 2 are heterozygous. And phenotypically, they're indistinguishable, but they have a difference in terms of how they cross for the next generation. All right, so let's talk about Punnet squares. So we just talked about how we assign the letters to the alleles. Basically, the dominant is going to be uppercase, and the lowercase is going to be the same letter as the dominant, but lowercase for the recessive. So again, we're not going to change from P to W. We're going to use P for both, 1 uppercase and 1 lowercase. And then we're going to determine the proportions of all the gametes that come out to be each of the different genotypes, and then the phenotypes from both parents. All right, so I'm going to skip through this and just kind of show you how that works out. So let's look. Here is the actual Punnet square. All right, so this is where we started. This was the original, this is the offspring of the original test cross. So this is going to be all heterozygous. So that means they have 1 copy of the dominant and 1 copy of the recessive allele. And when they're self-fertilizing, that means that half of their sperm and half of their eggs are each going to have 1 dominant and the recessive allele. So the eggs can be half dominant and half recessive, and the sperm, same thing, half dominant and half recessive. And that's denoted here by purple and white. And then when we cross them together, we're basically going to, in this square, look at the egg and the sperm that contributed to that square. In this square, look at the egg and the sperm that contributed to this square. In this square, look at the egg and the sperm that contributed to this square. And then here, same thing, egg and the sperm that contribute to that square. So you're basically going to look at the alleles that are on the edges and pull them together in these individual squares. And that gives us a phenotypic ratio of 3-to-1. What does that mean? That means that we have 3 of them are going to come out purple, and 1 of them is going to come out white, right? But this one is going to come out, so again, 3 of them are going to come out purple, and 1 of them is going to come out white. But this one is going to come out as homozygous dominant, and these 2 are going to come out as heterozygous. So while 3 of them are purple, phenotypically we have a 3-to-1 ratio. Genotypically, we have a 1 homozygous dominant, 2 heterozygous, and 1 homozygous recessive. So genotypically, we have a 1 to 2-to-1 ratio. Does that make sense? All right, so how do you find out if the purple plant that you're dealing with is this purple plant or this purple plant, right? How do you know which one it is? So we do something called a test cross. And a test cross is used to deduce if an organism, again with the dominant phenotype, in this case purple, is homozygous or heterozygous for that dominant allele. And basically what you're going to do is you're going to cross it with a homozygous recessive organism, right? If you cross it with the white plant, then you're going to know in the next generation. Because if you have the homozygous dominant, you're going to have all purple, right? So if you cross it with the homozygous dominant, the next generation is going to be all purple because this one can only make purple sperm and purple eggs, and those are always going to dominate, right? But if the phenotypic organism, which is the dominant phenotype, right, so it's purple, is heterozygous, genotypically, means half the offspring would be of the recessive phenotype. So we're going to have a 1-to-1 ratio of purple to white. So what does that look like? Okay, so this one is unknown. It's either homozygous dominant or homozygous recessive, right? This one, it's known. We know that all of the eggs are going to be recessive and all of the sperm are going to be recessive, right? So if we do a test cross, we are either going to see that 100% of them are purple, and that would be if we have homozygous dominant phenotype, all the sperm are going to be purple, all of the eggs here from this guy are going to be white, that's going to be, all of them are going to be heterozygous. It means they're all going to appear purple. If they're all purple, the unknown must be what Mendel was calling a true breeder, right? Or capital P, homozygous dominant. But if we get 50-50, that's because we start here, right? That means that we have 50% of the sperm are going to be purple, 50% are going to be white, so 50% dominant, 50% recessive. When we cross them out with that recessive, we're going to end up with 50-50 of a heterozygote and the homozygous recessive, right? So this is called a test cross. So Mendel used this to help pursue more complex questions, right? So now that he knows how certain things work and he knows the mathematical proportions, then he can ask questions relative to the inheritance of multiple traits. So how are these traits inherited together, etc. And so this is something called a di-cross or dihybrid cross, and we can look at two different traits. So for the one that I'm going to show you, we're going to be looking at seed color and seed shape. But there are multiple other traits that Mendel studied. So before I show you that dihybrid cross, I apologize, I want to show you several of the different traits that Gregor Mendel studied. Now, I'll remind you, he wanted to study only things that were dominant versus recessive. So only two different phenotypes. There's no range, right? There's no in-between for any of these. And he looked at things like seed shape, seed color, pod shape, flower color, the flower location, right? Is it found at the junctions or at the tips of the branches? The plant size, is it tall or dwarf, etc., and many, many, many more. So he had tons of books of data that are somewhere stored, I'm sure, in someone's archives that started the mathematical modeling for what we now understand to be genealogy and how genes are going to be passed on from one generation to the next. Okay, so for this dihybrid cross, we're looking at the color and the shape. And for the purposes of this, yellow color is dominant to green color. And smooth shape is dominant to wrinkled. Now remember, the dominant color gets the letter. So yellow is dominant, it gets capital Y. Green is recessive, it gets lowercase y. It does not get G, right? It gets lowercase y. That just shows you that these two are going to be an allelic combination that work together in a recessive dominant format. And in the same way, smooth being dominant gets the capital S, wrinkled being recessive gets the lowercase s. And we have a individual that is going to be smooth and yellow and is homozygous dominant for both of them. So this is our parental generation. And also another individual that is wrinkled and green, so homozygous recessive for both traits. And we know that this plant can only produce dominant alleles. It can only produce gametes that are capital S, capital Y. And this plant here can only produce gametes that are recessive alleles. It can only produce lowercase s and lowercase y. And so that means that the F1 generation consists solely of individuals that all have the same genotype. That is, they are heterozygous for both smooth and yellow. So heterozygous for both smooth skins and yellow coloring. And if we take those individuals and self-fertilize them, that is heterozygote by heterozygote, then we end up with a ratio of 9:3 to 3:1. And I'm not going to read it out to you. I'm going to go forward to this slide here. So if you were to look at this set of eggs, or the set of, peas, and allow them to self-fertilize, the eggs could be one of 4 different options, and the sperm could be one of those exact same 4 different options. So 1/4 of them are going to come out being dominant. And likewise, 1/4 of them are going to come out recessive. And 1/4 of them are going to come out recessive for one and dominant for the other. And then the reverse, right? So this one is going to be dominant for smooth and recessive for the color. And then we're going to see the exact same, and then the opposite here, right? So this is going to be recessive for smooth and dominant for color. And then here we see the exact same thing written out with the sperm. And if you're going to take all of these alleles and pull them together, so here we take 2 capitals and 2 capitals and pull them together, we end up with capital. So we end up with homozygous dominant for both traits. If we start at the opposite side and pull them down, again, we're starting with recessive on both sides, we end up with the exact opposite. We end up with homozygous recessive. Only 1 out of all 16 ends up homozygous recessive. However, if you look at the amount that end up being homozygous for, that are demonstrating the dominant phenotype for both, you're going to end up with 9. And if you end up with the ones, counting the ones that are demonstrating the dominant phenotype for one but not the other, for example, green but smooth, versus yellow but wrinkled, you're going to end up with 3. So dominant phenotype for both is 9. Dominant phenotype for green is 3. Dominant phenotype for yellow is 3. And recessive phenotype here, being the wrinkled and yellow, is going to be 1. Again, that's going to end up being a 9:3 to 3:1 ratio of each of these different phenotypes. So this is what a dihybrid cross looks like. Now you will have to do this for the laboratory section, and we're going to draw it all out for you, but don't worry, this is not going to be specifically a question that you're going to find on the exam. All right, so when Mendel did this, he actually found that if he's looking at the smooth versus wrinkled, he's finding a 3:1. And if he's looking yellow versus green, he's also finding 3:1. So he's still finding that 3:1 ratio that he was seeing with the yellow versus white pea plants, or the flower color. But if he's looking at them as the individual traits, he's getting the 9:3 to 3:1 ratio. And so that's going to lead him to believe that these traits were inherited independently, right? That you could, by random chance, get any combination. And that's called independent assortment. Basically what that says is that these 2 or more traits can be inherited independent of one another during meiosis. So if multiple traits are inherited independently, and that's because the alleles of one gene are distributed on totally different coin flip, basically, than the alleles for the other genes. But that would not be the case if they were on the same chromosomes. So it's only going to be the case if they are controlled by genes on different pairs of homologous chromosomes. Right? And the way that the homologous pairs line up during meiosis kind of helps to defend this strategy, because when the homologs line up on top of each other, there's going to be a random assortment and a random basically going to one side or the other randomly. So my point is that it could orient itself, in this case, it could orient itself top-bottom or bottom-top, right? It could flip over for each one of these. So you have a lot of different random ways that it's lining up during meiosis that allow them to pull independently of one another. And that allows for the gametes of the S and the Y, right, to separate independently. So you have independent assortment while you're creating your meiotic creations, right? So you end up with these specific allelic combinations. So 4 equally likely allelic combinations occur during that meiosis because of the independent assortment. Again, this is if these are alleles on different chromosomes. What's interesting is that Mendel actually published his work and then went unnoticed for quite a while, all the way until the 1900s. His work was so far ahead of his time that it was just raw math and nobody was there to be able to validate it, and so it didn't really get perpetuated until in 1900, several different biologists independently rediscovered the principles of inheritance, and then all at once started giving credit to Mendel. So all of a sudden he had no citations for his work, and then after 30 years he ended up being spurred to the forefront, being the person who laid the groundwork of genetics. All right, so the way that Mendelian inheritance works is based on a couple of different parameters that we believe to be true. 1, each trait is completely controlled by a single gene. 2, there's only 2 possible alleles of each gene. 3, one allele is completely dominant and the other is completely recessive. And this is just not the case for everything, right? Most traits are actually influenced in a much more varied and a much more subtle ways. So in order for Mendelian inheritance to work, all of these have to be true, and generally not all of these are true. So we can end up with things like incomplete dominance, where we have a phenotype that is in the middle. So the heterozygote is going to be an intermediate phenotype between the phenotypes of the homozygotes. For example, height, right? Very tall parent and a very short parent can end up with an average height individual. And so that's called incomplete dominance, and that means that we're not necessarily going to be dominant or recessive, that we're going to have an interplay between the two of us. And that actually is a lot more common than it was originally imagined. And again, that's called incomplete dominance. And an example of incomplete dominance is human hair texture. So if we have 2 different alleles, and they're both incomplete dominance, and we're going to call them H1 and H2. And if you have H1 and you had 2 copies, so if you're homozygous for H1, you have curly hair. And if you're homozygous for H2, you have straight hair, but if you're heterozygous, you have wavy hair. So you have an intermediate phenotype that's neither curly nor straight. And if 2 people with wavy hair, who are both that intermediate phenotype, right, that means that they're both heterozygote, their children could have any of the 3 hair types. Right, that looks like this. So here's a wavy-haired mother and a wavy-haired father. They can both create, so they're each H1, H2. They're heterozygote with an intermediate phenotype. They can each create sperm and egg that are either H1 or H2. And if we combine that down in this Punnet square, this individual has 2 copies of H1, this individual has H1 and H2, this one has also H1 and H2, and this individual has 2 copies of H2. That means that we have 1 individual with curly hair, 1 individual with straight hair, and 2 individuals with a wavy hair. Now, of course, these are percentage odds. That doesn't mean that every individual with 4 children is going to have this exact offspring, but those are just going to be the way that your percentage odds are going to play out, which is one of the things that worked out very well for Mendel because he had really high what we call N, or the number of individuals that he looked at, and so his statistics were very, very tight. But I digress. Another one of Mendel's characteristics is that a single gene had just a single allele, right? But oftentimes, multiple alleles of a single gene will exist. And although an individual can only have 2 different genes, right, because you only have 2 different locations to host those genes, a species can have multiple alleles for a given characteristic. And again, each individual is still only going to be able to carry 2 alleles for the characteristic. And an example of multi-allelic inheritance are human blood types, right? So human blood groups produce blood types A, B, and O. And there's several alleles in the system, 3 alleles, A and B, which together are what we call codominant. So they're both as strong, and O, which is recessive to either A or B. So these are both dominant over O, but together they're basically they're as strong, so they're codominant. And that is because alleles A and B code for an enzyme that adds a sugar, a glycoprotein, so to the end of these glycoproteins that protrude from the red blood cells. And allele A means that they don't add any sugar molecules at all. And from these 3 alleles, we can have 4 different blood types arise: A, B, O, and also AB. Again, A and B are dominant over O. So if you have the AA or the AO genotype, your blood type is going to be A. So these 2 are going to be indistinguishable because they're both going to have the A sugar on the outside. People with BB or BO genotypes have blood type B, and people with OO genotypes have the blood type O. Right? So AB individuals are going to have both A and B allele, and they're going to produce both types of enzymes. So they're going to have both. That means that the plasma membranes of their blood cells have both A glycoproteins and B glycoproteins. And that's going to be codominance, right? When heterozygotes express the phenotypes of both, that means they have both A and B glycoproteins, that's called codominance. So there's no intermediate. They're literally expressing both of them at the same time. Now, in addition to this, it gets a little more complicated because we were just talking about the glycoproteins on the surface of the blood cells, but in your bloodstream, you make antibodies against the glycoproteins that you lack, so the glycoproteins that you don't have. So if you have type A blood, you won't make type A antibodies, but you will make type B antibodies, and if you have type B blood, you'll make A antibodies. And if you have O blood, you make antibodies to both type A and type B, but if you have type AB blood, you don't make any antibodies at all. Say that 10 times fast. You don't make any antibodies at all. There we go. So these antibodies can cause red blood cells to bind together and rupture. So if you have antibodies that match up with the antigens, right, the surface markers on the red blood cells, this can cause a coagulation reaction. And so you have to be very careful to match your blood type very carefully before blood transfusions are made. Okay, but that doesn't mean that you can only transfer type O to type O and type AB to type AB and type A to A, right? And then we'll get to that in just a second. So type O blood has no sugars on it. So it's not attacked by antibodies in A, B, or AB blood, and that's why it's considered to be a universal donor. It can be transfused safely to everyone. However, this individual with type O blood can only receive type O blood, cannot receive A, B, or AB. So even though he can give it to anyone, he cannot receive it from anyone except his own blood type. And we don't really worry about the antibodies in the blood itself because it dilutes out very quickly, so it doesn't actually cause any problems in the recipient of the transfused blood. Now, if you have type O blood, again, you produce A and B antibodies, you can only receive blood from other type O donors. So what does this kind of look like? So if you have blood type A, you have 2 different options for what your genotype can be. You can either be homozygous dominant, AO, or you can be homozygous dominant AA, or you can be heterozygous, AO. Either way, you're going to express that A glycoprotein and end up having plasma antibodies against B glycoprotein, which means that you can receive blood from an individual who has A blood or O blood. Basically, you can receive blood from any individual that has no B glycoprotein. And additionally, you can donate to A or to AB. This just shows you the frequency in the United States. Here you have BB or BO. Again, it's going to have B glycoprotein. Going to have antibodies against A glycoprotein, which means that they can receive blood from B or O, but not from A or AB. Okay, so they can donate to B or AB, but they can't receive from AB, to be clear. Now, AB blood has both A and B glycoproteins, which means it can receive anything. It has neither glycoproteins in their plasma antibodies, so it doesn't have antibodies against A or B, which means it can receive blood from anybody, AB, A, B, or O. So it's a universal recipient, and it can only donate blood to itself, basically, 4% frequency. And then O and it's shown as lowercase. Why? Because in this case, these guys are codominant, remember, but O is recessive. So this is the homozygous recessive phenotype, which presents with neither A nor B glycoproteins on the surface of the red blood cells and has plasma antibodies against both A and B glycoproteins. So it can only receive blood from O, but can donate blood to any of them. And it's considered the universal donor with a frequency of 44% in the United States. Now, another thing that Mendel put down as a characteristic is that a trait could only be influenced by one gene, but multiple traits are actually influenced by many genes. And sometimes characteristics, instead of showing this discrete that he talked about, right, it's either one color or the other, it's either one location or the other, they actually are going to have continuous phenotypes. And some examples of this include height, IQ, skin color, body build, also grain color in wheat and in mice, coloration of their fur in mice. These traits are going to be influenced by at least two genes, but generally more genes, through a process called polygenic inheritance. And poly meaning many, genic meaning genes. So it's inherited, your phenotype is inherited from multiple different sets of genes. All right, so polygenetic inheritance means that the traits affected are often strongly, can be affected by the environment sometimes. Again, things like skin color, for example, which already has a wide range within the population, but then the environment can also affect it by, for example, sunning can change your skin tone, and that's going to further blur the differences among phenotypes. Now, as we just mentioned, two or more genes, but most of them are controlled by many more, including human height, which is controlled by at least 180 genes, and human skin color is controlled, that we know, by at least 3 genes, which are all paired up with incomplete dominant alleles. And incomplete dominant means that there's an intermediate phenotype. Again, this is just demonstrating that there's a wide range of skin tone because there's a lot of allelic frequency in human skin. Additionally, sometimes, this is another rule that breaks Mendel's patterns of inheritance, single genes often have effects on multiple phenotypes. So if an allele has multiple phenotypic effects, that's called pleiotropy. Say, for example, it influences a number of different gene loci. And a good example of that one, for example, gentlemen, the Y chromosome really doesn't carry much on it except male differentiation and development. So it has a gene called the SRY gene that encodes a protein that activates multiple other genes all in the male differentiation and development cascade. And that's going to stimulate the development of gonads into testes, etc. And ladies, if you don't end up with the expression of this SRY gene, that means that you're going to have the female sex by default. But that also means that you have an entire second X, which means that you're not necessarily susceptible to sex-related disorders, but we'll talk about that in just a little bit. But my point is that some genes, like the SRY gene, have an effect on multiple different gene loci, like many different sets of genes that, again, are involved in male differentiation and development. So sometimes, like, one light switch can influence many different gene loci. Additionally, just to complicate things, the environment can also drastically influence the genetic expression of certain, so you get a different phenotype. A good example of that would be Siamese cats. So a Siamese cat actually has a genotype for dark fur all over its body, but if your body gets too warm, 93 degrees or higher, the enzyme that produces the dark pigment is going to get turned off. So when kittens are inside the uterus, they're in the all-encompassing warmth. That means the enzyme is inactive and they're born completely pale everywhere. So they don't look like Siamese cats at all. But then over time, as they are, the ears and the nose and paws become exposed to cooler temperatures in the rest of the body, the dark pigment is then produced because the enzyme is not denatured. And that's going to show you the characteristic that's the pattern that's characteristic of the breed. See, so these guys here don't look anything like mama. Why not? Because the regions of mom's skin that are exposed to cooler temperatures are going to have the enzyme not deactivated, so still active, meaning that we're going to end up with the darker color. But these guys came fresh out of mom's uterus, so they had the enzyme inactivated. oftentimes genes on the chromosome are inherited together. So that would be called gene linkage. And when we talked about independent assortment, that only works if the loci are on different pairs of homologous chromosomes. If they are on the same chromosome, they're very likely to be traveling together, right? They're lined up on the same place of the metaphase plate. And so therefore, they're going to not be separated at anaphase 1. That means they're going to travel together most likely, and that's called gene linkage. A good example of genetic linkage is flower color and pollen. Right? So purple flower color is dominant to red, and long pollen shape is dominant to round. And so what you would expect would be that 9 to 3 to 3 to 1 ratio if they were inherited together, right? Your independent assortment would yield the gametes of 1/4 of homozygous dominant, 1/4 heterozygous with the P being the dominant trait, and then 1/4 heterozygous with L being the dominant trait, and then 1/4 homozygous recessive. Instead, almost all of the gametes are either homozygous dominant or homozygous recessive, and that indicates that they're traveling together and there's no chance for them to separate out. What does that mean? That means that they're on the same chromosome. So that means that the purple allele and the long allele are on the same chromosome, and this one here, the red allele, and this one, the round allele, again, on the same chromosome. And so when they separate out during the mitotic during the metaphase plate, they're actually separating to opposite sides. But interestingly enough, this actually led to the knowledge that genes that are on the same chromosome don't have to always stay together. So if we go backwards here, there's a lot of space between this gene and that gene. And as you recall, when we talked about meiosis, there's crossing over events that happen quite often. And so occasionally, the exchange of DNA between the sister chromatids means that the genes that were on the same chromosome might switch. And the further apart the genes are on the chromosome, the more likely crossing over is to occur between them. And sometimes, as we mentioned, these crossing over events happen 2 to 3 times per chromosome. So if the genes are on loci very far apart on the same chromosome, they often appear to assort completely independently. You can't even tell that they're gene linkage. So gene linkage is really only going to be something that's visible if they're really closely related on that chromosome. And that's because as many gametes as are produced within the genes are going to be exchanging, these crossing over, right? And that's going to make multiple different combinations of these alleles. So that genetic recombination occurs in prophase 1 of meiosis, and again creates new genetic combinations. And so if we're looking here at these sister chromatids with the purple and the long, and you're expecting these would probably assort together, but instead we have crossing over occurring 3 different locations between purple and between this allele and that allele, well then we're going to end up appearing to assort independently, even though they would actually be linked genes. Again, this is what crossing over looks like. So all of a sudden this purple is going to go with the short allele, and this recessive red is going to go with the long allele. Which means that we're going to end up with recombinations, right? So totally recombined chromatids that are going to express new inheritance, right, units, basically. So these are unchanged and recombined chromosomes after meiosis 2. So these guys are going to be recombined, and these guys are the original. And so because we have this recombination event happening so frequently, it's going to appear as though they are actually assorted independently, even though they were originally on the same chromosome. All right, so let's talk about sex linkage. Ladies, you have 2 X chromosomes, and men, you only have 1 because you have that Y chromosome that I just talked about, having very limited genes on it that are really only related to male differentiation and development. Now, although they are totally different, the X and the Y do link up during meiosis. They have a region called the pseudo-autosomal region, or the PAR, that allows them to link up during meiosis so they look like they are connected. But the rest of the ones that are not going to be our sex chromosomes, those are called autosomes. So they are going to be chromosomes 1 through 22. All right, again, the X and the Y chromosome look very, very different. But there's a region whereby they pair up, so it appears to be the same during mitosis. But the X is much, much larger than Y. Why is that? Because Y only carries information for becoming a dude. All the rest of the information that's carried on the X is not carried on the Y. So there's a lot of information that the X carries that the Y does not. Again, Y contains genes for male development, and as it turns out, it's not vital. If you don't have the active SRY gene, even if you have the Y chromosome, you're going to become a female. Or if you don't have the Y chromosome at all, you just have an XO, again, it's not going to be lethal. You're going to just have a female birth. Again, in that case, you're probably going to end up with some issues with reproduction later on in life, but it's going to be a viable defect. The X, on the other hand, contains a ton of genes. Genes for color vision, hair retention, blood clotting, and many more. And what do you see about these? What really strikes your interest about these three genetic information? Well, color vision, blood clotting, and hair retention are all things that are that have issues more in men than in women, right? Male pattern baldness, for example. More men are colorblind than women. More men succumb to hemophilia than women. Why is that? Because, ladies, when you have two copies of the X chromosome and one of them has an error in it, your body preferentially shuts that one down. Now, we're going to have to shut one down anyway because we don't want to have double the expression of men, but we could randomly shut that one down, which is what normally happens if they're both normal. But if one of them is abnormal, the abnormal one is preferentially shut down, meaning that we have much less of a chance to end up with only black and white vision or with male pattern baldness, because we'd have to actually have two bad copies of the X, whereas men, you only have to have one bad copy of the X because you only have one copy of the Y, which means that you, with this karyotype, are what we call hemizygous. You are neither hetero nor homozygous. You have only one copy. So what's on that X is what you get. All right, so again, males are XY, females are XX, and the sex chromosomes carried by the sperm determine the sex of the offspring. Why? Because, ladies, you can only give the X. So no matter what, you're giving the X. And men, you're either going to give the X or the Y. If you give the X in the sperm, an X-bearing sperm is going to create a female, and a Y-bearing sperm is going to create a male. What does this look like? Again, ladies, you can only produce the X. Now, again, just like anything else, you can have X1 or X2, so you can have two different copies of your X, right? And men, you're only going to have one copy of an X and one copy of the Y. Now, if we have an X-bearing sperm, we're going to end up with female offspring. If we have a Y-bearing sperm, we're going to end up with male offspring. Please keep in mind that these X's are not necessarily the exact same. They do carry the same genes, but just like any chromosome, there's allelic variations upon them, right? So genes that are carried only on one sex chromosome but not the other are considered to be sex-linked. And again, males, you are hemizygous for these genes. That means that you only get what you get. You don't have any compensation, right? And women, you are going to have two copies, which means that you're able to shut one down. You're going to shut one down in each cell. The X chromosome is much larger than the Y. It has over 1,000 genes. But that Y chromosome, much, much smaller, carries 78 genes, and those genes are pretty much only involved in the male differentiation and development cascade. Again, that is all going to start from SRY, so that SRY gene is going to set in motion all of these other 78 genes, which are going to induce the entire male developmental pathway, which means that if your SRY is normal, you end up with a male gender 100% of the time that you have the Y chromosome. However, if you have issues with the SRY or with any other part of that developmental cascade, sometimes you can have a Y chromosome and still present as female. Very few of the genes on the X chromosome have a role in female reproduction, right? So female sex is just kind of the default sex. Most of the genes on the X chromosome have no counterpart on the Y chromosome. So again, there's nothing that lines up with them. And oftentimes these X chromosomal genes are important to both sexes. Again, color vision, blood clotting, etc. And if the X and the Y have very few genes in common, that means that females, you can be homozygous or heterozygous for whatever characteristic is carried on that X, but men, you're hemizygous. That means you only have one copy, and what that copy says goes. Right? Males with only one copy of that X chromosome, you have no second copy to miss whatever recessive allele might be present, so you fully express any X-linked allele that you have, whether the allele is dominant or recessive. So there's no way for it to have any interplay. Example of this is red-green color blindness. It's very common in the male population. It's caused by a recessive allele on either of two different genes located on the X chromosome. The normal allele, we're going to call it C with a capital C, just like you would normally have a dominant and recessive relationship. It's going to carry for color. Encodes proteins that allow an eye cone to be sensitive to red light and another sensitive to green light. So you have different sets of eye cones that are going to be sensitive to different types of light. Now, if we have a recessive allele, and there are multiple different types of recessive alleles of these genes, and again, we're just going to blanket call them lowercase c. That means that the individual who's afflicted cannot distinguish between red and green. So while a man can have a genotype of capital CY or a lowercase cy, that means he's hemizygous for either the dominant or the recessive allele. It means he either has a color vision allele or a non-color vision allele on his X chromosome and no corresponding gene on the Y. So he gets what he gets, and that is it. Right? Ladies, however, if you are homozygous dominant, obviously you'll have normal color vision, but you could be heterozygous or homozygous recessive. This heterozygous is something that the men cannot be. And in a heterozygous individual, that means they can still have color vision because they have one good copy of that C, right? For men, it's one or the other. You either have normal color vision if your X carries the normal C, capital C allele, or be colorblind if your X chromosome carries the lowercase c allele. There's really no in between. But a woman will only be colorblind if she has two bad copies of the C. Now, a man who is colorblind, it's depicted as lowercase c. Why? Because this is the X that's carrying the lowercase c. Will pass his defective allele on to his daughters, only to his daughters. Why not his sons? Because his sons are going to get the Y, right? Same thing with male pattern baldness. You have to, it only skips a generation because it goes into the ladies. It does not get passed on to the sons directly, right? And that's because the sons are going to be getting the Y from their father. Now, a heterozygous woman, even if she has normal color vision herself, it could possibly pass her defective allele onto half of her sons. So half of her sons will end up with colorblind alleles and the other half will not. Here's just a quick way to demonstrate the difference between what normal color vision looks like and red-green color blindness looks like. And if these two look the same to you, then it's very likely that you're afflicted with some sort of red-green color blindness. But here we have the red scale, and this is the green scale. And as you can see, the red and the green in a colorblind individual are going to appear to be approximately the same. And then this is just showing the expected outcomes if you were to cross a female parent who has heterozygote, so she has one normal color and one abnormal color X, and the male has the normal color vision. So if he has a normal color X and a Y, that means that in the female offspring who are getting the normal color X from the father, it doesn't matter whether they get the normal color X or the abnormal X from their mother, they're both going to have normal vision because they have at least one good copy. So in a female, being a heterozygote means you're still going to be able to see color vision. But in a male who receives that, who gets the Y from his father, he does not have any compensation from a normal X, which means that this individual here is actually going to be colorblind, this male individual here. Alright, so a lot of human diseases are influenced by genetics. So we've taken records over many generations when we're looking at the medical history of particular families who carry specific genetic traits of interest. And those are called pedigrees. And if we take records extending back many generations, you can show the genetic relationships between related individuals and the disease that they carry. So pedigree analysis is often combined, especially these days, with more modern technology like molecular genetics to determine gene action and expression. So if you know who carries which traits, then you can try to figure out how those traits are expressed. And so because of that, scientists now know which genes are responsible for sickle cell anemia, for example, hemophilia, muscular dystrophy, Marfan syndrome, cystic fibrosis, and many, many, many more. So what does a pedigree look like? Well, pedigree looks a little bit different for a dominant trait and for a recessive trait. All right, so for a dominant trait, everybody who has the trait also has the gene, right? So they're all going to be filled in. Let me back up a second. Males are going to be squares, females are going to be circles. This indicates parent and offspring from those parents. So this would be a male-female relationship that ended up with 2 sons and 2 daughters, right? And as you can see, you would look at the offspring. So this individual would have, we have one affected individual, marries a non-affected individual, and ends up with, we have 1, 2, 3, 4, 5 offspring. We have 2 affected and 3 unaffected. This individual, we have 1 affected and a wife that is not affected, and we have 1, 2, 3, 4, 5. In this case, we have 3 that are going to be affected. So approximately half of the offspring are going to have the affected trait, and they're going to show the trait if they carry the trait, right? There is no carrier. Whereas in this recessive trait, these guys here would be considered carriers, and what that means is that they are a known carrier for that trait, that they don't express that trait, but their offspring could, again, if they're going to cross with another carrier. So here's the mom and dad combination who are both carriers, which means that their offspring are very likely to be either ill or carriers, right? So that's what a pedigree looks like. And again, this is generation 1, generation 2, generation, so great-grandparents, grandparents, parents, offspring. So multiple traits are going to be inherited in a simple Mendelian fashion. So although I just gave you all those exceptions to the rule, a lot of things are carried that way. Things like cleft chin or freckles, right? And so most of the traits are actually controlled by a single gene with a recessive and a dominant allele. But some disorders are caused by defective alleles of the single gene, and they can be inherited either as a recessive trait or as a dominant trait. So new alleles are produced by mutation constantly, and usually they code for non-functional proteins. So usually they're no big deal and there's no change in the individual, and it's not even something that shows up. But sometimes there is a change that does end up being passed on and creating functional proteins or changing the function of a protein. Usually alleles that code for non-functional proteins are recessive to those that code for functional ones. So just like we talked about when we talked about the purple flower versus the white flower, basically the purple flower is going to be a functional protein. It creates a purple pigment. And the white flower has a non-functional protein. It doesn't create a pigment. So the absence of pigment is going to be easily compensated for by the presence of pigment, which means that if you have one allele that creates pigment, you're going to be purple. So the presence of one normal allele can oftentimes be enough to generate enough functional protein that the heterozygotes are phenotypically indistinguishable from the homozygotes with the two normal alleles, which means that the trait that you're of distinguishment only really shows up, like the white flower color, when you have the homozygous recessive. That means that you can have heterozygous individuals that can be carriers for the recessive genetic trait, but have a normal phenotype on the outside. Also, recessive genes are more likely to occur in a homozygous combination if individuals who are related have children. So the inbreeding, right? So the closer you are related to the individual that you're breeding with, the more likely that a recessive gene might become a homozygous recessive trait. Right? So close relatives are more likely to have the same recessive alleles and so are more likely to produce whatever that homozygous recessive phenotype is. All right, so let me talk about being albino for just a moment. So albinism happens when there's a defect in melanin production. And again, a defective protein is always recessive to a productive one. So melanin is a pigment that colors the skin cells, and it's produced by an enzyme tyrosinase. And the allele that creates tyrosinase is TYR. TYR encodes a defective, if it encodes a defective tyrosinase protein, that means that no melanin is produced. And you're going to make a condition called albinism. So if humans or other animals are homozygous, again, a heterozygote is going to produce enough pigment to be able to have a normal phenotype. But if you're homozygous for TYR, you have no color in your skin, your fur, or your eyes. So your skin and your hair are going to appear to be very light. The eyes are going to appear to be pink. Oh, that didn't show up. Well, there should be an image of an albino rabbit right here. So I apologize, but use your imagination, I suppose. All right, so let's talk about sickle cell anemia. Sickle cell anemia is caused by a defective allele for the synthesis of a protein called hemoglobin. Hemoglobin is important in transporting oxygen. It's located in red blood cells. And if we have a mutant version of this hemoglobin, it causes the molecules in the red blood cells to stick together. So they take it on a crescent shape, which looks like a sickle. It also makes them really easily broken. And it allows for blood clots to form, and it can cause oxygen starvation in downstream tissues and paralysis and necrosis. And this condition is known as sickle cell anemia. Now, if you're homozygous for the sickle cell allele, you're only going to make defective hemoglobin and therefore only sickled cells. And you're going to be very, very sick. But heterozygotes have about half normal and half abnormal hemoglobin, so they normally have only a few sickled cells, and they're generally not seriously affected. And usually only people who are homozygous for sickle cell show symptoms, so it's considered to be a recessive disorder, although the heterozygote does kind of have its own phenotype. And what's interesting is that it affords some protection to malaria, which is why it has persisted for so long. So especially in sub-Saharan Africans and therefore in African Americans who are direct descent from sub-Saharan Africans. 8% of African Americans here in the United States are heterozygous for sickle cell anemia. And the allele is actually very rare in Caucasians. Why? Because the individuals who lived in Africa were exposed to malaria, and heterozygotes confer a little bit of resistance to the parasite that causes malaria. And so that means that it has a high percentage in African American populations, where it has a rarity in heterozygotes of Caucasians. And that corresponds directly with the rarity of malaria in northern climates. And again, this has to do with the ecosystem, where we don't have a ton of mosquitoes in northern climates, / they end up freezing over the winter and don't survive, as opposed to someplace like Africa, where they don't have freezing over the winter, perhaps. And so the mosquitoes, which are going to be bearing these diseases, don't end up having any sort of dying off season, basically. All right, so this is what normal red blood cells look like on the left. This is what sickle red blood cells look like on the right. You can see they are drastically different in shape. They're prone to clumping. They're very fragile. They break easily. And if you're heterozygote and this is all you have, that can be lethal very early on. You're going to need constant blood transfusions. Now, a dominant disease is basically one that can, that if you have one bad copy, you're going to have the disease. So instead of having a recessive disease like we just talked about where you have to have two bad copies of the allele, here you only have to have one bad copy. And it can be passed on from parent to offspring as long as the parent lives long enough to reproduce. Dominant disease alleles can arise as new mutations occasionally in the DNA of eggs or sperm of unaffected parents, leading to the rise of offspring who are affected. But occasionally it does pass on if the parents are able to survive to reproduce. And when we look at dominance of alleles, so the dominant versus the recessive allele, there's a lot of different mechanisms that create that allelic dominance. So sometimes dominant alleles encode an abnormal protein that is basically stronger than or interferes with the function of the normal protein. Sometimes dominant alleles encode proteins that carry out toxic reactions. So they may damage the environment, or an allele could be dominant if it creates an overactive protein, or if it is active at inappropriate times and places. So if there was regulation on the normal allele, but there is no regulation on the abnormal protein, then you can end up with a protein that kind of runs aberrant. Now, an example of a dominant disease is Huntington's disease. Huntington's disease is caused by a defective allele that creates a defective protein that kills brain cells in specific brain regions. And it happens slowly. Over time, it's a progressive deterioration of parts of the brain that results in loss of coordination, flailing movements, personality disturbances, eventually death. And the unfortunate part is that generally it doesn't become clear or manifest until adulthood, meaning that it can be maintained in the population because generally you've already had your children by the time that you have had this problem. So the gene encodes for a protein called Huntingtin, which has unknown normal function, but mutant Huntingtin seems to interfere with the action of normal Huntingtin somehow and cause these large aggregates in nerve cells, which ultimately leads to the death of these nerve cells. All right. We also have sex-linked disorders. So we just talked about sex-linked disorders we talked about hemophilia. And basically that's what happens when the X chromosome, which again has many genes that have no counterpart on the Y chromosome, when there's only one X chromosome in the male, they have no other allele available to exert dominance. So therefore, they're going to end up with whatever allele they have presenting as the allele that's presenting the phenotype. So sex-linked diseases occur more in males than in females. And they have a unique pattern of inheritance. So because the son receives his X chromosomes from his mom and passes it on to his daughter, right? So the gene doesn't ever exist on the X chromosome. So he's not going to pass it on to his sons, and he's not going to receive it from his dad, which means that sex-linked genes are going to skip generations because the affected male typically carries it, sends it on to a carrier daughter who doesn't express anything, but then bears affected sons. So it often will skip a generation. And there's several defective alleles for characteristics encoded on the X chromosome that are known to mankind, including red-green color blindness, hemophilia, etc. Hemophilia, again, it's a recessive allele on the X chromosome, and it's going to cause a deficiency in a protein needed for blood clotting. Now, if you're a female and you have two X's, then you can end up with minor blood loss, but you have some normal gene protein, so you are going to perhaps be susceptible to anemia and bruise easily, but you're not going to end up bleeding to death the way that men do. So if you look at the hemophilia gene in Queen Victoria being passed along the royal families of Europe, you see that quite often the females are carriers. They may show some signs like they have delayed. They might bleed a little bit longer and delayed healing, increased bruising, that sort of thing. But the males end up highly affected, right? Which means that they have to be very, very careful because a small cut from slicing through a bagel into their finger can end up leading to an emergency room, right? And again, what you see is that generally you're going to see that the females who are carriers pass that affected gene along to the males who are hemizygous and therefore have no good gene to be able to compensate for it. Again, you see males are mainly affected, females are carriers, although they do kind of have their own moderate intermediate phenotype. Okay, so sometimes we can end up with genetic disorders arising by the chromosomes aligning incorrectly and separating incorrectly during meiosis, and that's called non-disjunction. And non-disjunction means that we're going to create gametes that have one too many or one too few chromosomes. Now, usually this is going to be what we call embryonic lethal, or it's going to cause a spontaneous abortive event. But sometimes individuals that are going to be embryos that arise from this defective disorder, can survive to birth and beyond. Now, only if they end up with triplicate, so monosomy, meaning you only have one copy of your chromosomes, is never going to be viable. But triplicate or trisomy is sometimes viable depending on which chromosome you've replicated. And so this is what happens during normal meiosis, right? During normal meiosis 1, you're going to pull each of the sister chromosomes to opposite sides, and they're going to end up being pulled apart during meiosis 2. So we end up with 4 genetically distinct, but again, they're all haploid individuals. Now, if we have non-disjunction during meiosis 1, we end up with this whole set going to that side, which means that we end up with half of them are going to end up with N+1, so an extra chromosome, and half of them are going to end up with N-1. And again, a monosomy is not going to be tolerated, whereas a trisomy may be tolerated. And then this one here, we're going to end up, if we have non-disjunction during meiosis 2, so if the non-disjunction event occurs here, so we've had a normal split here, but not here, then we end up with these guys, again, they'll have a normal, so these will be normal N, but these will be N+1 and N-1. So depending on when the non-disjunction happens or when the chromosomes fail to align properly, you can end up with all of the gametes with plus and minus 1 or half of the gametes with plus and minus 1. Either way, all of the minus ones are going to be non-viable. Now, if we end up with non-disjunction of our sex chromosomes, we can produce abnormal numbers of X and Y chromosomes. And these are interesting because these are generally going to be viable. As long as you have one X, it's generally going to be viable. Why is that? Well, if we have extra X's, we tend to shut them down the same way we do our secondary X. So if we have 2 X's, like normal for females, one's always shut down anyway. But if we have 3 X's, for some reason 2 shut down, and 4 X's, 3 shut down. So it seems to be pretty well tolerated to have extra X's. And if we end up with no sex chromosomes, it can be O sperm, so you can end up with like XO. And XO is viable, but YO is not. Again, without the X, you are going to be doing very poorly. So if you end up with YY sperm or XY sperm that are going to meet up with a normal egg, you can end up with what's called trisomy. So non-disjunction of sex chromosomes in females can produce eggs that are O or XX instead of eggs with one X, whereas men can create XXXY or YY. Now, if those abnormal gametes fuse with normal gametes, then you can end up with a zygote that have normal numbers of autosomes but abnormal numbers of sex chromosomes, and that leads to several normal abnormalities. Again, you'll notice that all of our abnormalities include at least one X. YO is not viable. Why? Because the Y doesn't have anything on it except male differentiation and development, and a lot of the stuff on the X is very important. So this XO, this is called Turner syndrome. This is perfectly viable. XXX, same thing, perfectly viable. In fact, XXX and XXY are going to present. This is going to present like a normal female and like a normal male. One of these is going to get shut down. Two of these are going to get shut down. And then XYY, they actually are going to be a little bit more aggressive. This, I believe, is called Jacobson syndrome, if I recall correctly, and it does lead to an increase in testosterone, and they actually end up incarcerated at higher rates than the regular population for violent crime because the Y doesn't get turned off, so you end up with double the amount of Y characteristics. Let's see. So the genes on the X are super essential for survival. So any embryo without at least one X is going to spontaneously abort. So again, YO is just not going to be viable because you need at least one X chromosome to survive. XO is going to be Turner syndrome. When a female has only one X chromosome, usually XO females don't undergo puberty properly, so they don't end up menstruating, and they fail to develop secondary sexual characteristics, so they tend to appear to be underdeveloped. And you can produce hormone treatment to help promote physical development, but they're never going to produce mature eggs, so they're generally across the board going to remain infertile. Additional symptoms of XO syndrome include short stature, neck folds around the folds of skin around the neck, and also cardiovascular disease, kidney defects, and deafness. They only have one chromosome, so women with Turner syndrome are just as susceptible to recessive disorders like red bean color blindness, hemophilia, and male pattern baldness as men are. Another genetic disorder that can arise can be trisomy. So trisomy X would be if we have 3 copies of the X, and that actually, as I mentioned, results in a normal fertile woman with an extra X chromosome. In fact, they show no abnormal symptoms and typically don't even know it until some sort of reason arises for them to get a genetic test. There is a higher chance of learning disabilities and a tendency towards being extremely tall associated with trisomy X for some reason, and for some mechanism that prevents that extra X chromosome from being included in their eggs, Women with trisomy X, that's generally going to be totally fertile, and they're going to bear normal XX and XY children. So it's a situation where it's going to right itself during its own meiotic processes during the next generation. However, if men end up with trisomy, so you can have XXY, they're going to have an extra X chromosome. Again, you're typically not going to show any symptoms, although sometimes you can have some secondary sexual characteristics like partial breast development, or broad hips, smaller testes, etc. They're generally going to be infertile because of low sperm count, but that doesn't mean that they're impotent, so they are still able to reproduce, and oftentimes with IVF, they're going to see just fine reproductive events. Typically, diagnosis of XXY are actually made when the individual seeks medical attention because they're having issues with infertility. So they have no other secondary symptoms, and they didn't realize that they were carrying the extra X until they have an issue with either repeated fetal loss events, etc. Now, if a male has trisomy with XYY and they have an extra Y chromosome, they tend to have extra high levels of testosterone. They often have severe acne and height, extremely height, often can be very hairy, lower voices. It occurs about 1 out of every 1,000 males. It's also sometimes associated with an increased likelihood of learning disabilities, but that's very light. And they also tend to be incarcerated at higher rates than the general population, generally due to violent crimes associated with increased testosterone. So this is just an overview of everything that I just told you, so I'm not going to go back through this again. But if you want a summary, this is a table that summarizes different effects of non-disjunction during meiosis and the different disorders associated with that. So non-disjunction of autosomes can also occur. So I just showed you non-disjunction of your sex chromosomes, but non-disjunction of autosomes can also occur during meiosis in either the father or the mother, which can sometimes result in eggs or sperm that are missing an autosome or that have 2 copies of the autosome. And if the eggs or the sperm are missing an autosome, they're typically not, they're single copy autosome embryos are going to abort very early in development. These are not viable. So a fusion of these gametes with a normal sperm or egg is going to end up in a zygote with either 1 copy or 3 copies of the affected autosome. Again, the 1 copy, they're not going to survive. In fact, most of the 3 copies are also not going to survive. Most of the embryos with 3 copies of an autosome are also going to spontaneously abort. However, there are 3 separate chromosomes that are viable in a trisomy. So a fraction of embryos born with 3 copies of either of these chromosomes, chromosome 13, chromosome 18, or chromosome 21, survive to birth. 13 and 18 are generally going to be lethal within the first couple of years of life, but trisomy 21 can lead to a relatively normal lifespan, although a little bit shortened lifespan, and they do end up being infertile. Now, the frequency of non-disjunction increases with the age of the parents. It's a maternal aging effect, and so the older a woman is as she goes to procreate, she ends up with an increased risk of trisomies. Now, the trisomy that you're probably very familiar with is trisomy 21, that's known as Down syndrome. That affects individuals who have 3 copies of trisomy, or of chromosome 21, or trisomy 21. It occurs 1 out of every 800 births or so, and again, it is associated with increased maternal age. And symptoms of Down syndrome include distinctive physical characteristics like a weakened muscle tone, a smaller mouth that's held partially open at most times because the tongue is enlarged, so the small mouth can't accommodate the large tongue, and distinctively shaped eyelids. Also characterized by a increased risk of infection and heart maladies, and also varying degrees of mental retardation that can range to the very severe. They also tend to be infertile, at least men do tend to be infertile, although there are cases of trisomy 21 females who are fertile. And this here is showing you a karyotype, so a normal karyotype would show 2 copies of every chromosome. And here's a karyotype of trisomy 21 showing that 3rd copy of chromosome 21 right there. So I do know that that was a very long lecture. I really appreciate you sticking with it. So aloha, see you next time, and happy studying.